Awesome. Thank you for the kind introduction, Erica. It's an honor to be presenting at the KI Image Awards ceremony. So my name is Arnav Chhabra, and I'm an alum of the Koch Institute. I actually used to be a postdoctoral fellow in Dr. Sangeeta Bhatia's lab there. So at the Bhatia lab, we're a team of engineers who like to derive inspiration from biology. As an example, we'll take a novel scientific finding and leverage that discovery to develop a tool that will either help us with early detection of disease or help us improve an actual therapy. The clinical problem that really motivates us is highlighted in this graph here. For people with end-stage organ failure, their only option is a transplant. However, the supply of these organs is very limited. As you can see, the organ waitlist has rapidly increased over the last 20 years, while the number of transplants and donors has not kept up. To bridge this gap, many researchers, including our lab, is utilizing cells as therapeutics. But cell therapies suffer from one major problem. They need blood vessels to function well when they're implanted in the body. The blood vessels represent the highway that transports nutrients and oxygen to all of its cells. In these images, I'm showing what happens when you don't have a blood vessel in a mass of tissue. As you can see in the left image, the cells in the center start to die due to a lack of nutrients and oxygen. While there are many different ways to think about this problem, we actually look towards cancer to inspire us. So cancer cells are incredibly pesky. As they grow and invade your body, they produce these signals to recruit blood vessels to help sustain themselves. So we thought to ourselves, can we leverage the same mechanisms, but to help form blood vessels instead inside of our cell therapies? We looked at many different ways to do this. And after a lot of research, we actually arrived at a very interesting method. As it turns out, you can electrically stimulate tissues in a dish to help form blood vessels. And by using electrical signals as a stimulus, we are able to form these blood vessels in an extremely controlled way. We can detect areas of the tissue that are low in oxygen and nutrients and stimulate those regions to promote the formation of blood vessels. And the result is as follows. So to create this image, we used electrical fields to fabricate a network of blood vessels, which are shown in the red, around engineered mini livers, which are shown in cyan. The resulting vasculature efficiently transports nutrients to the mini organs, promoting their long-term survival and demonstrating the potential of electrical stimulation to improve both transplant technology and cell-based therapies. We think this approach has a lot of potential to improve cell therapies and in turn improve outcomes for patients suffering from end-stage disease. And with that, I'd like to acknowledge a whole host of people. I'd like to thank my project partners, Dr. Edward Tan, who's a postdoc in the Bhatia lab, and Keval Vyas, who was a technician in the Bhatia lab. I'd also like to thank my entire lab, specifically my PhD and postdoc advisor and longtime mentor, Dr. Sangeeta Bhatia. Lastly, I'd also like to thank Erica for running the Koch Image Awards and the 2021 judges for selecting our work.